session, and I want to say thank you to the organizers of this symposium for putting together such a great series of talks. So today I'm going to be talking about some work I've been doing in Mimulus guttatus. So as an evolutionary biologist, I'm fascinated by the amount of variation present within and across plant populations. And one trait that is especially important in today's series is plant fitness. And as many of you can probably relate to, it's sometimes really difficult to measure fitness of an individual plant, especially when there are many metrics of fitness, or if you want to draw or make comparisons between individuals that may have different fitness components. And that's really relevant, I think, to today's talk, as I mentioned, as we talk about sort of translational research from herbaceous plants into the realm of yield of agricultural systems. And I think this quote by Harper really sums up kind of the foundation of my talk, which is, if the plant is only the means by which the seed produces more seed, the variations in the efficiency of this process are extraordinary. And so I think this really lays the groundwork for the amount of variation present in plant populations in both traits like fitness, but also things like life history strategies or life cycle variation. So today I'm going to share with you some work I've been doing in the Mimulus guttatus system, where a large amount of life cycle variation is present. And some of these fitness components are a little bit more straightforward to measure, things like overwinter survival, survival to reproduction, and other aspects are a little bit trickier. And specifically, I'm going to talk about two aspects of fecundity selection, or fecundity fitness. The first being sexual reproduction, or the number of seeds produced either through selfing or outcrossing events, and also asexual reproduction, or the production of genetically identical clones. And many plants reproduce through both of these routes. And I think a great foundation question for this particular series is, why sacrifice immediate reproduction for perennial growth? And to look for answers to this, we can look at the foundations of life history theory, where this paradox has been resolved by proposing that optimal life histories are a function of both fitness, or fecundity, reproductive output, but also the relative survival of individuals at different life history stages. And this has great applications to agriculture. And I think a big question, if you're interacting with land users and farmers, is how do you convince individuals that reduced early season yield is beneficial in the long term? So to increase things like survival in future years, or future yield within that first year, but also subsequent years after that. And so today I'm going to address three questions. Does fitness vary by parental genotypes and their F1 hybrids? Do genotypes differ in sexual and asexual components of fitness? And does fitness in year 1 affect fitness in year 2? And to answer these questions, I've been using the model system, Mimulus guttatus, which exists in two broad ectopic forms, perennial and annual. So perennial plants are found in environments which are continually wet. So the plants have continual access to water, and therefore have continual vegetative presence, or presence of these asexual stolons. The annual environment is drastically different, where it's seasonally moist or seasonally wet, fed by a large input of water into the system from snowmelt or early spring rain. And there's a high premium on early flowering. So annual and perennial populations, shown here in red and blue respectively, co-occur through the entire wide range of the species, from Mexico all the way through the Alaskan archipelago. And if we look at the phylogeny, we don't see a strong pattern of two major clades, which are annual and perennial, despite a single origin of annuality. But if we recode this phylogeny, there's a much stronger pattern, which is that of isolation by geographic distance. And so what you can see here are 4 major groups, and at the end of the phylogeny tips are open and closed circles. And you can see that annuals and perennials are interdigitated throughout the entire phylogeny. So individuals are more related to the perennial and annual populations nearest to them rather than across that single origin of perenniality. And to sort of exploit this phenotypic variation for our study, we selected an individual population from each of these major groups. And so today I'm going to be talking about the parent populations. The first is the northern perennial population from the island of Haida Gwaii off the coast of British Columbia, an annual plant that invests very little into asexual growth and reproduces primarily through seed production from Vancouver Island, an individual population from the coastal perennial clade, and another perennial that's more inland. And in order to generate both phenotypic and genetic diversity, we took an approach that's really common in agricultural settings as well. And so individuals from two populations were inbred to homozygosity and then crossed together to generate a heterozygous F1, or a hybrid. We then grew up the F2 generation following the selfing of the F1 and intercrossed these plants down to the F4. So they're all outbred, intercrossed F4 mapping populations, and there are three distinct populations that share a common maternal plant. And so all of the progeny from each of the three crosses is derived from the northern perennial, serving as the maternal parent, being crossed to either the annual, the inland perennial, or the coastal perennial. We then packed up, went to Vancouver Island onto the native range of this plant species, and grew out individuals of the parents, the outcross parents, the F1s, and then 300 individuals from each of the three mapping populations. And we measured a suite of traits. Today I'll be focusing mainly on components of fitness, but we also measured things like flowering time, leaf size, plant architecture and branching patterns, clonal growth and biomass and fecundity in year one. We measured overwinter survival into the next year, and then remeasured things like clonal biomass and seed production in year two. So I'm just going to walk through quickly how we measured and how we're estimating sexual fitness. So we counted all of the flowers in the entire population, which was about 300,000 in year one. We then examined individual fruits to get a plant-based estimate of how many of them actually set seed. We then collected seed pods from every individual plant and counted the number of seeds present, and that's giving us an estimate of total seed number per plant. So we're going to move on to what we found so far, and this is again focusing on only the parents and the F1. So I'm going to walk you through this graph because it's going to be the way I present the results for the entire talk. So our trait will be on the y-axis. On the x-axis are the 7 genotypes that we're looking at. NP, northern perennial, furthest to the side in black, is our maternal genotype. The remaining 3, CP, IP, and A, are the paternal genotypes. And then in between them will be the F1, representative F1s of each cross. And so what we can see here is that the northern perennial invests the most resources into clonal growth, and so into these sort of perennial growth patterns. The coastal perennial and inland perennial fall sort of intermediate, and annual plants have the lowest investment into clonal growth, which is what we expect because seed production is their primary means of reproduction and fitness output. But we see a really interesting pattern when we overlay the F1s on here, in that the F1s all show the dominant maternal phenotype. So despite being crossed to 3 separate populations, each of the F1 hybrids have the same investment into clonal growth as the maternal parent. We see a very different pattern for fecundity, or seed number. And so we see that TSG, the maternal parent, invests the smallest amount into seed production, but all of the F1s show a pattern of heterosis. And so the seed number is highest in the F1s relative to either the maternal or paternal parent. And if we look at these two fitness components together, we can see that there's not an apparent trade-off, as one might expect, between sexual and asexual reproduction. In fact, these hybrid F1s are good at both components of fitness. So they produce both a large number of seeds and also a large investment into clonal asexual growth, which is related to overwinter survival and fitness in year 2. And so clonal growth in year 1, shown on the bottom of both axes here on the X, is related to overwinter survival. So investment in year 1 into clonal growth facilitates higher overwinter survival rates, and it also facilitates higher seed number in year 2. And data that we just got in last week from this last field season is you also see a similar pattern. So individual genotypes that have high clonal growth in year 1 also have high clonal growth in year 2. And so to wrap up, the 3 questions I talked about today with the parent populations and the F1s. First, we see that F1s show either dominance or heterosis in 2 components of fitness. We see that the northern perennial has the greatest fitness through clonal reproduction. The inland perennial has greatest fitness through sexual reproduction. And in general, the annuals fare poorly in terms of both investment into sexual and asexual reproduction. And I should point out that despite the fact that our field site was in a very perennial-like habitat, so extremely wet, the genetic basis to seed production in the annual was not plastic enough to sort of take advantage of that higher water environment. And lastly, we see that clonal growth is associated with overwinter survival, and it's also associated with higher fitness in year 2. So the work that I shared today is just a small amount of the work that we have going on with this project. So if you recall, I also mentioned that we have mapping populations out over the 2 years. So here you're seeing distributions of 2 components of fitness in the mapping populations, fecundity and clonal growth. There's a wide range of variation present in each of the mapping populations, and we're currently working to genotype the individuals from all 3 F4 mapping populations to identify genomic regions involved in sexual and asexual components of fitness, and to characterize allelic contributions to fitness and dominance relationships. So with that, I'd like to acknowledge Jannice Friedman, the members of the Friedman Lab, and the dozens of field assistants I had on Vancouver Island helping me collect and count hundreds of thousands of flowers and seeds. Carol and Stu Carwithen that generously allowed us to set up an entire field site in their backyard. So to plow over their agricultural field and build an entire field site, weather station and all, from the ground up, and funding from the National Science Foundation and Syracuse University. And with that, I would love to take any questions that you may have.
So this is all in a relatively well-watered environment. Does this lack of trade-off hold up in a relatively unwatered environment?
Right, so this is about as well-watered of an environment as you can. Our field site sits on top of a natural spring where we were wading through water all the way into August. And so yes, I would predict that this trade-off between sexual and asexual reproduction would be much more present in a more stressful environment. And we've done greenhouse experiments to examine this that we're looking at the data for.
With the advantages that you're seeing in the F1, do you predict perhaps a breakdown in an F2?
Yes. So I think that what we're seeing here is, the relationship at this sort of phenotypic level, right? And so we know that there are many genes underlying these fitness components, and many of them probably do not have that same dominance interaction that we see. And so I think we are going to see a large amount of breakdown in the mapping populations as we start to sort out those alleles and pair them into different combinations. And I think F1s are kind of a standard approach for a lot of agricultural systems. And I think that for sustainability, it's important to understand how stable these interactions will be as we advance these generations. Because it's not necessarily the most efficient thing to be continually regenerating F1s.
How often do you see natural hybrids between like for example, perennial and annual populations, and do you ever see natural hybrids exhibiting these heterosis phenotypes?
I personally have not seen this. It's something I'll have to look into. We do see a large amount of plasticity in these traits, and so whether those are hybrids in nature or just environmentally driven phenotypes, I can't speak to that.
Thank you.
Yeah, cool. Right, thank you, Matthew. That was an awesome talk.